My name is Martine Bos. I'm a molecular microbiologist and I've been working since 2.5 years at Microbiome. So far we have been mostly dedicated to developing tools, diagnostic tests in the medical field. So we do molecular tests to detect bacteria, viruses, fungi or any pathogen. So on the one hand, we can detect very specific types of bacteria. And on the other hand, we have developed a new technique, which is called RESPRO, which we actually use now in the RESFOOD Project. So that's a sort of a new development for our company. Now we are entering the field of food quality, water quality, which is very different from medical applications. But this technique, this RESPRO technique, sort of measures complex microflora. So we can look in any kind of sample when there are many types of bacteria present that you don't know of. We can make a whole profile of that. And that is the strength of this new technique. And so the RESFOOD Project, we look at water quality. We can see all the bacteria present in the water, have some idea about what types of bacteria. We can also assist in looking at the effect of disinfection. So make a profile before disinfection and make a profile after, and then determine whether the disinfection worked. We basically predominantly address the microbiological food quality and safety. So looking at all kinds of bacteria. And of course, one of the interesting aspects I think of us entering in this project is we don't know much about the role of bacteria on food, in food quality. We of course know some of the pathogens that we don't want there. They're pathogenic for humans. But there are of course also many good bacteria that need to be there, that may even beneficial. So now for the first time I think we get a sort of a first glimpse of everything that's present there. We don't know yet how exactly to interpret it, but we definitely would be interested to investigate further in this project if we can find out more about the role of not the pathogens, but also the good bacteria. The detection and monitoring technologies we use are all based on DNA. So the classical microbiological methods were all based on culture. So you need live bacteria, and also you need to know where the bacteria grow on, because we now know that many bacteria we cannot grow on the plate. We just don't know what the conditions are for them to grow. But if you start with DNA, you can always measure DNA. Bacteria don't need to be alive. And also going from DNA as a starting material means that you can measure in a very sensitive way. Because basically you need only one molecule of DNA, so one bacterium basically. And then we do this reaction called PCR, polymerase chain reaction, that you can amplify this DNA until a great amount of material, and then it's easy to measure. So all our tests are based on this PCR. Food safety is of course very important. We've seen many examples in the past of things going very badly wrong with E. coli. And I think it's not only for the people that are infected by the E. coli, but also for the growers and the people that process food. They have very bad consequences of such an event. So I think it's very important to improve on that. And I think first by measuring what is actually there. And the second thing in the RESFOOD Project is that we actually take that knowledge to develop monitoring devices to be able to immediately take action whenever a pathogen is present in the whole chain. So I think that really will improve food quality.